Okay, so in today's lab, I want to talk a little bit about what a grant proposal is, why we need to think about what a proposal is and why people write them, and what they provide for scientists and the scientific community. So if you are interested in any kind of career that deals with research, this actually applies for physicians in the medical field too. So you at times in your career may have to write what's called a grant proposal. So when you come up with an idea, an observation, or you have identified a problem that you want to find a solution to, normally you just don't have money lying around to buy the reagents, to buy the giant equipment, to pay scientists to do the work for you, and you can't just do it by yourself. So there are there are big, giant funding agencies. Some are small, some are very large. And these funding agencies, they want to put money into your project. They're kind of like investors. And so your job as a scientist, if you're writing a proposal, is you want to make it sound logical. You want to make it sound relevant. You want to make it sound plausible. So you want to make it sound innovative, something that hasn't yet been done before. You want to provide an idea for an experiment that will produce data that no matter what the data is still going to be new, informative data. And that's kind of the tricky part of grant proposals. And so you present this proposal to this funding agency. They probably get tons of proposals. There's grant proposal season. And so they basically just pick the project that they feel has the most promise for a problem that is worth solving. So, a typical grant proposal is going to consist of a little bit of work that you've already done, and this will help you build an argument for the plausibility of it. You can say like, hey, here's this major problem. Here's why it's a problem. Here's why we need to solve it. And so far, no one has done it. Here's what we currently know about this, or here's our current solution, and it's kind of crappy. So we have already done a little bit of our own work just to see if our idea would even work in the first place. And so it looks like so far so good. We have this data and it's very promising. So we're ready to embark on the next step, but we need your help. We need financial support. And then the proposal will consist of 3 to 4 different experiments that will help the funding agency decide like, wow, they've really thought about that experiment. They've thought of the controls. They've thought about like what could go wrong, and they have a backup for what could go wrong, or they have alternatives for this. And no matter what their data is, it's still going to be new, exciting data. So even if the data's no change, that's still the way that you set it up should be very exciting. And this can kind of come down to your writing approach. So it's like, are you a good salesman? If so, you can probably make anything sound exciting. So that's a big part of writing the proposal. So you're going to be doing this in a group. You've already selected your teams, and you will stick with that team for the rest of the semester. And in terms of what's happening in lab class, this is our last organized lab where we all log in at 10 a.m. Next week, I'm just hosting team Dr. B meetings. So a whole team will come at a very specified time and you'll present to me your aims. The more detailed you have that experiment planned out, the more feedback I can give you on the spot. If you don't have a very much detail, I can say like, sounds like it could be good, so good luck. But if you have more details, I can say like, have you thought of this? Or how many mice do you want to use? Or how long are you going to do that? What concentration are you going to use? I can ask you things like that. So you're going to make the most of the meetings, and you are going to be the leader of the meetings. You're going to show up, present stuff to me, and if I have questions, I'll ask you. So today's our last opportunity to really make sure that we understand what a proposal is, understand what the assignment is asking of us, and make sure that we have the details that we need so that we can write a cohesive, concise proposal. Okay, so we can go ahead and get started. Now, I provided to you a scenario, and I just want to be very clear that this is fake. I did not really go to the Amazonian rainforest. There are, to my knowledge, no plants that are fluorescent at night. Maybe there are, but not to my knowledge. So it's not a real plant. I made up the species name of plant. And, but GFP as a protein really does exist in real life. So I'm kind of just using some imagination, but the idea of formulating an experiment can definitely be done with the fake information that I'm giving you. Okay, so in the first breakout room, in the first breakout room, you are going to make sure that you fully understand the background of this scenario. And so to do that, really you want to identify what the problem is. So what do you think the problem is? And also, why is it a problem? Usually grant proposals in the biomedical research field typically have something to do with some kind of disorder, disease, condition that we currently don't have a solution or a treatment for. So that kind of like gives you a huge clue as to what the problem is. And so you don't only want to make clear what the problem is, but you also want to make sure that the moneyed people know that it's important, right? So if it's something that only affects like one person that happens to have 3 eyeballs, maybe they're not going to give you $5 million to come up with a solution how to remove that 3rd eyeball. Like, yeah, I mean, I get it's important for that person, but like how many people does it really affect? So what is the problem? Why is it important? And so you kind of want to do this from 2 perspectives. You want to do it from the perspective of just humans in general, non-scientists, okay? And then you want to kind of make sure that it seems significant for the scientific field. So we'll just call it the scientific community. And that's basically what I'm asking you to do is this is how I'm saying it's kind of similar to the lit review, because you want to catch anyone's attention. And then you're going to start trying to hone in on the nerds. Like, okay, nerds, this is obviously a problem. So, but let me tell you why it's a problem, right? So more of the cell molecular part of things. Okay. And then any kind of background information. So if you're going to bring up, so this is easy enough. So what is the kind of disorder or the disease that this whole scenario focuses around? So just go ahead and put it in the chat. This is an easy one, big deal here. Great. Okay, so we know that we're going to be talking about skin cancer. I'm going to leave it up to you and your group to decide if you are going to focus on a specific type of skin cancer or you're going to leave it broad, but whatever it is, if you're talking about skin cancer, that means you're talking about a disease that happens in the skin cells, okay? So there are different types of skin cells. You might want to just list some of the skin cell types there are, just like there's different types of neurons in the brain, there's different types of skin cells in the skin. If you are talking about cancer, you might want to talk about the cell and molecular basis of cancer in skin. So what are some of the, what is cancer in the skin? What is actually happening? What are the cells doing wrong? And are there any genetic bases for that? So what is being misregulated? And that can be broad in terms of cell biology, so cell processes, or it can be, or it can be detailed in terms of molecular interaction. So what molecules, what genes are being disrupted, misregulated, etc. So this would be types. Okay, and if you're talking about cancer, is there a specific type? And then this is another easy one, but just to make sure that you talk about it in your groups. What is the proposed solution? What is the thing that we're hoping will cure skin cancer? Yeah, okay. So this whole proposal focuses around this magic molecule called GFP. And so I am telling you that it does not really exist in this plant, but it does exist in real life. Does anyone know what organism really does have a code a genetic code for GFP. Yeah. So GFP, since we're talking about a completely different organism, but I want to make this as authentic as possible, what we're going to say is that the G-funk, GFP is highly conserved or highly orthologous, the sequence of it, to the GFP gene from jellies. Aquarius, Aquarius, something like that, Victoria. Okay, so if you're going to provide some background on GFP, jellies, correct, yes. See, I didn't say jellyfish, and I'm not an animal biologist, but I only know that because I've been reprimanded before. Okay, so I learned from my mistakes. So yes, you want to use that as your reference. So the idea is if GFP gene from A Victoria and that of G-funk are orthologous, that means their sequence is very similar. There's a lot of homology in the sequence. Now, if that's the case, then you can predict or hypothesize that the GFP protein from both of those organisms predominantly or may function and be shaped and structured the same. But these are just hypotheses based on the sequence homology. So if you're going to provide a background on GFP, you got to make sure that you're mentioning that it's the information that you're providing is from what is known about A Victoria GFP, not G-funk GFP, because that's actually what you're doing. You're trying to figure out what does G-funk GFP do? How is it structured? What kind of molecular interactions can it participate in, etc. Okay, cool. So these are some of the things that you can think about when you're talking about the background, but not only that, don't forget about the problem itself. Why is that significant for just the human population? Okay, so I think 15 minutes should be good, and then we'll come back to the class and talk about it. So before I send you into breakout rooms, are there any questions? Okay, so okay, so we are making a list of the things that kind of describe skin cancer as the problem of this potential grant proposal. So I'm just going to kind of skim these chat contributions. So it looks like we have something along the lines of hard to detect, okay, hard to detect early. Okay, it's very common, that's good, right? You're collectively making skin cancer a relevant or a big problem that needs to be solved. So it's the most common type of cancer. There's a lot of stats that people are putting. So what are some of the stats that could impress the non-scientists, not just, 1 in a million people that get it? Okay, and so usually you are going to be writing to a United States grant funding agency. So I think stats have to be relevant to the country that you're trying to get money from. Okay, so it's common and it's malignant. Okay, great. All right, so people are talking about the causes. I'm gonna leave that. Maybe I'll just draw a little line right here and maybe the relevance to scientists, the fact that UV rays can be the cause. You can write about this in a non-scientific way, or you can talk about it or talk about it with a little more scientific depth, because we just finished talking about UV rays and the kind of radiation they are. So let's just keep that in mind for now. Okay, so I'm going to put UV here, and I'm going to also put UV here, because if you are going to talk about them in both, it's not like an actual section, but it probably could be a different paragraph. You're going to want to talk about that differently. Okay, so common, so more stats. Okay, so quality of life. Great. Alrighty. Great. This is a good one. So why is this important? Because we currently do not have a good treatment. So currently, and this is like a very important proponent of a grant proposal, because if you are proposing a new solution, it needs to be significantly better than the one we already have. And the one that we already have better not be that great. If you want money. Of course, we want a good solution, but you know, if we have a treatment that works great, there's no reason to make another great treatment. So I think it's awesome that somebody said that currently the current treatment we have, we'll call it unsuccessful, or we can call it inefficient. And there is a definite difference between treatment of and prevention of. So I don't know if anyone wrote that, but you know, I think that kind of goes in line with what is up here, hard to detect early. So it's, if we don't know how to detect it early, then it's really hard for us to prevent it from happening. But I would go and probably assume that we don't have no current preventives. So these are kind of like the big problems here, okay? So skin cancer is the problem, but what's the bigger issue is that we don't have a solution to the problem. Okay, great. Cancer is common. Okay, genetics or external, great. So I'm gonna put genetics may also go in both kind of categories. Okay, awesome. I think this is a really good list for the background of skin cancer that will capture the attention of the non-scientist. Okay, so let's do that same thing. This time, try to put like a cell molecular spin on it. So in this situation here, so let's go back up here. This is where we're gonna maybe start talking about using words that a non-scientist may not be familiar with. So again, just phrases, don't press enter. I'll give you 20 seconds, 30 seconds to do that, and then I'll have you press enter. So go ahead and type in the things that you've talked about in your groups. All right, and go ahead and press enter. Okay. All right, this is good. Okay, so let's do like cell types. And I'm just writing a few that I'm seeing as I'm scanning down. So I'm seeing stuff like melanocytes. I'm seeing stuff like epidermal cells. So when you're writing this in your grant proposal, it might require maybe like a couple sentences just to talk about the different cell types and you know how they should normally function. Let's see. Okay, basal cells, squamous cells. Okay, I'm just gonna write a few more. Basal, squamous. Okay. And then cancer types, skin cancer types. I'm seeing stuff like squamous cell carcinoma. Do I see that or am I just making that up? I thought I saw it. Okay, so we have melanomas and non-melanomas. Right, let's see. Okay. Okay, so it looks like melanoma is the specific type that people are, oh, basal cell carcinoma. Okay, so most people are writing melanoma. Some people wrote basal cell carcinoma. They're both skin cancer cell types. So you as a team have to decide what kind of cancer you're going to focus your proposal around. Squamous cell carcinoma, okay. So here's my first big piece of advice. All these cancers are important, but you have to pick one of them, and your entire proposal is going to kind of center around that. So if you pick the melanoma, you have to have a reason for picking that one versus the other one. Maybe you have a really great model, or maybe that affects more people, or maybe the symptoms of this are worse, or the prognosis is worse. Oh yeah, we definitely want to talk about prognosis here. Okay, like what's the proportion of people that will survive and will defeat this kind of cancer? If it's low, then that makes it a significant thing, right? And according, because I'm thinking about it now, some other kind of stats, not just total number of people, but when you're writing a proposal, some of the other attributes that people want to know about is what age group, what kind of ethnicity, demographic, okay? And then doesn't impact one sex over another. Okay, so those are kind of the stats that you definitely want to at least mention. Okay, so up to you and your group, which of the cancers are you going to kind of go down the path of trying to solve? And then I think that now, let's see if there's anything else that I want to talk about. Oh, so we're talking about causes. I see a couple people saying pyrimidine dimer. Okay, cool. So let's see, maybe characteristics of cancer. Okay, so the characteristics include misregulated or unregulated. We'll just say poorly regulated cell cycle. Specifically, the cells are replicating their DNA and then they're dividing without checking for mistakes, right? So the DNA is copied with all its mutations with damage, and along the way, you know, if those mutations are in very specific genes, like genes that control the cell cycle, then those gene products are not functioning properly. So that means the cell is dividing when it's not supposed to, and then passing the damaged DNA onto that generation. And then that cell, it doesn't know that it's made wrong. So it replicates its DNA and it divides, and it just keeps going and going and going. So poorly regulated cell cycle. Some of the causes could be damage. The type of damage that you guys are writing is the potential pyrimidine dimers. And you're saying the cause of that could be UV, right? And so that's when we kind of bring this back up. So UV, any non-scientist knows or should know that the sun is the source of UV rays. As a scientist, maybe this is when you want to start talking about UV and the type of radiation it is and the effect that it has on the DNA, right? So we know that pyrimidine dimers are damage to the backbone. They're not changing the nucleotide bases. They're causing the bases to detach from their complementary bases and instead attach to one another via these covalent bonds. So instead of hydrogen bonding with bases on the other strand, they're now covalently bonding with one another, disrupting the backbone, and any kind of distortion to the shape of the DNA is going to interfere with replication and transcription. Okay, so let's think what else. I was reading or listening to somebody talk about this. I can't remember if it was like a friend or a podcast. But so pop quiz, what is the type of machinery that typically can fix the damage by UV rays? Come on, you can do it. This was just in the test on Friday. What repair mechanisms fix pyrimidine dimers? Not base. Nucleotide excision repair. Yes. Okay. So nucleotide excision repair, though, that's a bunch of proteins that they have to recognize the damage, they have to remove the damage, and then they have to, you know, put in the correct stuff. So we don't just have one set of machinery because, you know, we're going out in the outside world almost every day. Quarantine doesn't mean stay inside. It just means kind of stay away from people. So we have multiple sets of this machinery, and I forget the actual rate of how fast that they can fix it, but I think it's like, oh yeah, the nucleotide excision repair machinery fixes the damage by UV rays at about 300 pyrimidine dimers per second or something, or maybe per minute or something ridiculous like that. But if you have overexposure to the sun, like let's say it's 90 degrees out and you're like, base tan, here I come, going to the beach, that UV damage is probably on the order, I think it was like 5-fold. So 1,500 pyrimidine dimers, and the machinery, it's not that machinery is broken, it's that the machinery is working so fast, but the amount of damage is so much more, and it just can't fix all of that in the amount of time. So I thought that was really crazy. So it's not a broken machinery, it's just you're over exposing yourself to UV. Okay, cool. So last, I think, major topic that I think we need to talk about with regard to the problem are the genetics. So when we talk about genetics, we're talking about heritability. We're talking about actual genes that are mutated. So maybe you have talked about this in your group, maybe you didn't, but let's see what people come up with. So again, another 20 seconds to kind of type into the chat and we'll see what we come up with. Okay, let's see what you have. Hit enter. Okay. All right. Okay, so we actually have some gene names. Okay, so we have CDK N2A, and then we have BAP1. Okay, so mutations in either of these genes increase your risk. Okay, so there are associated genes with melanoma. So these are melanoma genes. Okay, familial is far less common than spontaneous. Okay, so Joaquin is letting us know that usually people develop skin cancer because it's accumulations of mutations throughout a person's life, and they're less likely to have inherited it from their parents. So you would say spontaneous is more common than inheritable or familial. Great. So now we have some demographics. So we say Caucasians are more susceptible. Okay, so if Caucasians are more susceptible, that is something that you can write in the demographics part of this, but now we're putting a scientific spin on it. Why is because Caucasians have less melanin. So this may have something to do with the gene for melanin production. So a gene codes for melanin production. Melanin is the product. It's the protein product. Okay, and melanin is what protects you from the sun. Okay, we have some more, I would say these are molecular components. So we have our pyrimidines, so cytosine and thymine are pyrimidines, and they are the ones that are going to be involved in the pyrimidine dimers. Great. Okay, all right. Fair skin is okay, okay. Cool, great. So we've got even some stats for this too that you can find. Wow, really great job. Cool. So when you're thinking about the aim, the experiment that you're going to kind of focus on, there's a couple things that you need to decide. Are you going to be looking at a cellular process like the cell cycle? If so, what are some, here's a word from back in the weeks 2 and 3 of the Kikuchi et al. Paper, what are some markers for the cell cycle? Markers, remember, are some kind of gene that is like a clue that the cell is dividing, or that the cell is cancerous, or that the cell is a neuron. So you may want to consider, if our team is going to look at the rate of cell cycle, what are some markers? that we might investigate. If your team is going to go the route of looking at regulation of certain genes, or maybe repair of certain genes, or like fix the mutation of certain genes, maybe you're going to go to the genetic route, and that is okay too. So it's kind of one or the other, and I would not try to investigate both of those genes, but just one. So the idea here is not to create this complicated experiment with so many different variables that it's hard to keep track of. The goal of this for you at this stage in your scientific path is to come up with one very well coordinated, organized, developed experiment with proper controls, with the reagents that are going to allow you to answer your question, and research into how you're going to do that. So simpler is better, because simpler means that you're going to have fewer, they're called pitfalls. Like, well, did you think about that? And you're like, no, I didn't. How are you going to fix that? I don't know, I didn't think about that. So you want to develop an experiment where there are few pitfalls, but that you have a backup. Like, oh, if this antibody doesn't work, that's okay. I'm gonna not do antibody staining. Instead, I'll do a Western blot, and I'll just use a different antibody, or whatever it may be. So by doing the background like this gives you ideas of the route that you can take when you're developing your aim. Okay, so let's make sure we have hit everything up. Oh, we're waiting for one more thing, or we need to do one more thing. Last but not least, we need to have a little bit of information about GFP. So maybe you had time to do this, maybe you didn't, but go ahead and take 20 seconds. Anything that you did talk about GFP in your group, it could be, what does the name stand for? It could be the length, it could be number of X, whatever you talked about. So go ahead and type it in. Do not press enter. I'll give you a few seconds to do that. All right, let's see what you got. Go ahead and press enter. Okay, so we have what it stands for. It stands for green fluorescent protein. Okay, so we've kind of mentioned some of these things already. It has high homology. To a Victoria GFP gene. And that's really important to state in the background. Otherwise, people are going to say, like, why would you think it would work, or why do you propose these functions? Okay. Okay. Cool. So people are writing about how it actually makes things glow. All right. Okay. In a Victoria, this thing is 28 kilodaltons. So we're talking about size. 4.2 nanometers. That's probably the protein. So we don't really measure proteins in nanometers, but you can maybe find some information about the structure. Is it quaternary structured? Does it have common? So how else? So if you don't know this about GFP, and I just said, hey, here's a protein. What would you do to analyze this protein so that you can describe this protein? What are some of the features that you would look at, analyze, anything about a protein that we have talked about up until this point? So we've kind of already talked about function, right? So we know we know one of the known functions, and we're trying to figure out some more functions. But what else about proteins? What kind of information can you provide that might give a scientist a better idea of what the protein looks like, is shaped like, anything? How it might get into cells, out of cells. There we go. So you might want to describe its molecular properties. And that means, you know, is it traditionally hydrophilic, hydrophobic, polar, non-polar? If that's the case, are there regions where there's a lot of the same amino acids? Yeah, so structure and hydropathy. So structure, hydropathy. If we're talking about structure, are there any regions with a lot of alpha helices or beta sheets? Let's see, polarity. Okay, let's see what else. So that's the protein version of it. So this is all protein. And we also know that proteins are coded for by DNA. And if you came to lecture yesterday, we also know that a gene just isn't a piece of DNA, like there's other regions involved. And so we've talked about things like inverted repeats. We've talked about dinucleotide repeats and trinucleotide repeats. So you can actually analyze the sequence of the DNA in that aspect too. So are there any interesting features at the level of the DNA? Okay, so we know that it comes from a eukaryote. And so we know that the eukaryote genes have introns and exons and introns. So one way that scientists describe genes is by the number of exons there are, and if there is any like important features. Like, oh yeah, it has a span of CG repeats or CAG repeats. So you would want to look into that as well. And maybe that doesn't go not into huge depth into the background, but some of these details might go into your actual aim, depending on which of these things you're going to investigate. So I would say for the most part, however, everyone's aims are going to be dealing with the protein version of that, because you're going to be using the GFP to see if you can either cure or prevent skin cancer. Okay, and so the last thing that we kind of have to determine is the solution. And so we know that the solution is you're proposing to use GFP, but you need to decide as a group, do you want to propose to use it as a treatment, okay, or a preventative? Those are two completely different ideas. Okay. Because if we already know that there's some genes that when they're mutated lead to a higher probability of getting this cancer, maybe you would want to use GFP to reverse the mutations of those genes so that that person doesn't get cancer. That would be a preventative. A treatment is the person has already suffered some level of skin cancer and they have either lesions or whatever it is they may be expressing on their skin. And so you're hoping to use GFP as a treatment for those symptoms, or I guess reversal of the disease. So there's a couple different avenues that you can go down, and it's very important to decide on one and focus your experiment around answering that objective. You cannot say, GFP is going to cure skin cancer, because I'm going to ask, what do you mean cure? So cure the symptoms, cure the genetic mutations, cure the heritability. So this is going to require specificity. Okay, so let's kind of make sure we have everything here. You guys can all see it. Great. Yeah, so number of subunits, let's go to. Okay. Anything else that we should add to any of these categories that you want to make sure that I add there? Okay, Cool. Well, I think that that is good. So that when you're writing a grant proposal, the background is kind of like the intro-ish to a molecular, to a biomedical research paper or a report that you've kind of already written. But I would say it's more along the lines of the lit review, how you start your lit review, and then, but you're not going to be recapping all the stuff that's already known. You're just like kind of laying the foundation. Hey, here's the problem. Here's why it's important. Here's how this problem is initiated in people in the first place. And then here is an intro into some of the molecule that we're using to provide a potential solution. And then that's pretty much it. So I would say like a background, maybe it's going to be like a page, page and a half, maybe page and a half. And then you're going to move right into the preliminary experiments. And the outline that I provided on the document on Canvas really is, it just looks like that. You're going to have those subtitles like preliminary experiment, and then you'll say specific objective. So what was the objective of the preliminary experiment? You'll just go through each of those one by one, and it's chunky and choppy, and it's not supposed to be this nice flowing thing. The background should be a nice flowing paragraph to paragraph to paragraph. And then when you get to the prelim experiment, you're not writing material and methods. You're writing more like, here are the overall steps that have been done, and then you move right into your preliminary data. So a grant proposal, the preliminary stuff you've already done, doesn't have a material and method section. Even though some stuff was done, there's no material method section. So that takes kind of the weight off of that section. If you're like, how much detail should I add? It's really like, to do this, this was done. To do this, this was done. And you just keep going through one by one. Okay, so Rachel's asking current costs, and I think, yeah, this might go up here. Currently, the United States spends X number of millions of dollars on research for skin cancer. Currently, the United States spends X number of dollars on treatment, or an average patient spends X number of dollars. Yes, I think cost is also very important. Okay, cool. So I think this is great. This is good. So I think we can take a break at this moment. So let's be back ready to work again at 11:15. So it gives you a little bit longer than 5 minutes. And when we come back, we're going to be spending time in a breakout room, really kind of coming up with a flow chart. So this was a one of the questions on a homework assignment. So everyone should have this. And it's really, you basically are comparing notes and making sure that you understand the steps and the correct order of the steps, and just some of the important reagents. So don't just say an antibody is used. What antibody? Don't just say a restriction enzyme is used. Which restriction enzymes? And so we'll talk about that when we get back. Okay, so we'll see you in about 6 minutes. Okay, so let's see, the first major step that was done in the preliminary experiment was that, looks like everyone is pretty much writing this. Yeah, is right now you're starting with a plant, and you can't really do experiments with a plant. And you don't want to extract just the protein, because the goal is to do a lot of experiments. And so if you only have a small little sample of a plant, you gotta first build up the amount of, I guess, reagent that you need in order to do those experiments. So in order to do that, you're gonna extract not total protein, but total DNA. Right, so when we did the Western blot, we extracted total protein because we were doing one Western blot, or maybe we could have used that protein lysate for a couple Western blots. But after the contents of that tube was gone, it's gone. And we don't, here we have the possibility of growing more piece of tea loom, extracting protein again, etc. But in this scenario, we don't have that option because we were only able to bring a little bit of GFUNC back with us from the Amazon. And so we need to make sure that we have the source, which is total DNA. And the main approach really is some kind of extraction protocol that's very similar to the one that we use for proteins. You might just use a couple different concentrations of salt and whatnot so that you can extract or isolate or precipitate out the DNA versus the proteins. So a DNA extraction protocol, pretty basic. Okay. And the desired product, again, is pretty straightforward. You're going to have a liquid solution of GFUNC DNA. And remember, that includes all of the DNA, right? It's not just one cell. It's not just leaf cells. It's not just stem cells. It's all of the DNA, but the DNA is the same in every single nuclei of every single cell. So it's all chromosomes. There's no way to just have GFP at this moment. Okay, so people are calling it gangsta funk plant. Well, I gotta be real, that's kind of how I started with the name. So G-funk era with the gangster twist. Okay, so great. That is the first major thing. So I will draw these tiny little cartoons to make sure that now we're gonna do something. What do we do with this DNA? So again, let's go with the next thing. So what is the next? And I'm going to give you a hint here. There's actually, it's kind of two steps. And they're not dependent on each other, but they both kind of have to happen at the same time. So go ahead and write what the next two things are that have to be done. Don't press enter. What needs to be done? How do you do it? What's the desired product for each one? All right, go ahead and press enter. Ooh. Okay. So let's see. Ooh, people are getting ahead of themselves. Okay, isolation of GFP, isolation of GFP, isolation of GFUNC and linearization of plasmid. Almost. GFUNC is the plant. You're not isolating the plant. Isolating the gene, not specific enough. What gene are we talking about? Restriction enzymes are also very vague, but yes, we are using restriction enzymes. So as I'm asking these questions, you should be updating the answers in the chat. If I'm saying it's not specific enough, make it specific enough. Okay, so I have a restriction digest for PBAD. Okay, good. All right, so I think most people are, at least we know that in this tube here, we have all of the DNA and GFUNC is somewhere mixed into one of the chromosomes. So we don't want all of the chromosomes. We somehow just want GFP, gene. Okay, so remember the difference between genes and proteins. Genes, we're talking about DNA, RNA, and then protein is the protein product. So one of the things that needs to be done is you want to isolate the GFP gene from the rest of the chromosomes, from the rest of the DNA. So this is it comes from a plant. So plant is a eukaryote. Eukaryotes have linear chromosomes. So I'm just going to draw a couple chromosomes. So here's one chromosome. Here's another chromosome. And if I can zoom in, let's pretend that GFP is just one tiny little section of one chromosome. So let's see, I'll just kind of draw a square around it. So maybe this is the GFP gene. So you just want this. You don't want to try to replicate all of the GFUNC's DNA. You just want GFP gene. So isolate the GFP gene, and people said that you're going to do this using an approach called a restriction digest. Restriction digest. Other people said that to do a restriction digest, you need Restriction enzymes. Okay, there's hundreds of restriction enzymes. So which restriction enzyme are we using, or did I use? So you don't have to wait to press enter, but I am waiting for a specific restriction enzyme name. Where can you find it? In the documents that I posted. So the resources, the videos that I had you watch, the scenarios potentially in there. So I'll give you some time to kind of look through your notes, look maybe endonuclease, that's the type. So a restriction enzyme is an endonuclease. Okay, so it looks like people have some homework to do. So I'm not giving the answer away, but you need some names here. I gave you a name of a restriction enzyme, definitely in one of the videos. So it might have been in the introductory part of it. I'm not too sure, but my Tuesday class knew it, so I know I posted it. So you need the names of the enzyme. What I will tell you is I gave you the name of one enzyme. And then I'm going to give this piece of information to you, restriction enzyme number 2. You are going to figure out. That's one of the things that's actually due next week, is you're going to figure it out, which is why I had you download that application called Snap Gene. So today after class, it's actually already up on Canvas. I've posted a demo on how to use Snap Gene and how to design what restriction enzymes you want to use. It also shows you how to design certain PCR primers. So you need to look this up, because this information was provided for you. So a couple things to remember about a restriction digest is, yes, you do need to use restriction enzymes. Those restriction enzymes, the type of enzymes they are endonucleases. Okay, that's just a category, it's not a specific name. And these endonucleases, so when, this is a question that you don't have to wait to press enter, but when you say digest, what exactly is happening? So if you had to describe what's happening, what would you say? Okay, so a digest, okay, good. People are answering the question before I asked it. So cutting, yeah, then my next question was cutting what? And so yes, a digest means the restriction enzymes are cutting DNA. Cool. What does cutting mean? Cutting what? Cutting what part of the DNA? Okay, so we know it's going to cut at a specific palindromic sequence. So each restriction enzyme has one specific sequence that it recognizes. When it recognizes this, it cuts. It cuts the phosphoester bonds. Yes. Okay. Okay. So at a palindromic sequence, it's literally cutting, not literally cutting, it's hydrolyzing. Cutting means hydrolyzing the phosphoester bond in the backbone. Okay, that's what a restriction digest is using an enzyme. An enzyme scans the DNA until it finds its perfect, most favorite palindromic sequence, and it only has one. And it finds it, and then in that palindromic sequence, it's like, okay, once I find my sequence, I know I'm supposed to cut between these two letters, right? These two nucleotides. I don't cut the whole thing into smithereens. I just clip the phosphoester backbone, and I do it on both strands. So essentially, if you're cutting the DNA on both strands, you're basically cutting the DNA. You're creating a double-strand break, essentially. And now the two fragments can come apart. So if you think about it in terms of, let's say this is the huge chromosome of the plant, and let's say that this region right here is your GFP gene. You're going to use a restriction enzyme. Maybe this tiny little region here is the palindromic sequence for restriction enzyme 1. This is going to cut the phosphoester backbone of both strands. Okay. Notice I'm not cutting right at the border, because sometimes you can't find a palindromic sequence right at the start of the gene or right at the end of the gene. Sometimes it's a little bit further away, upstream of the gene. Okay, and then you use a different restriction enzyme if it's possible, so that you can, maybe that one cuts a little bit further downstream. And this is the palindromic sequence for restriction enzyme number 2. Okay, so if this is happening all in a tube, remember this is all happening in a tube, you add the restriction enzymes in, you add the appropriate buffer in, and you let the enzymes do their job. So at the end of this, if you could zoom in, what you would have is this tiny little chunk of the chromosome. And then you'd have the GFP completely separate, right? So there's your GFP, I'll highlight it so we know it's the GFP. And then we have the other part of the chromosome. That's all floating around in this tube. Not only that, but you have all the other G-funk chromosomes, right? So let's pretend G-funk has 10 chromosomes. That means you have all of these chromosomes floating around. And you don't just have one cell. Remember, you homogenized, you crushed up the whole plant. So you have tons of cells. So you have lots of copies of this. So this, to your eye, looks like it's clear liquid, but really you have a bunch of pieces of DNA floating in here from many different cells. Some of them are your G-funk gene. Okay, it's all in there, the whole combination of it. Okay, one thing I'm going to add to this little cartoon here is that these restriction enzymes, they don't leave an actual substance on the edges of the DNA, but the way that they cut it, some of the enzymes leave what's known as a sticky end, and it's not actually sticky. It's not like that, but it basically means that there's an overhang of a couple different nucleotides. So the bases are just sticking down, like, oh man, where are my complementary bases? I'm dying to base pair back up with another strand. They're just hanging out. They're overhang, not hungover. Okay, so this will leave the same overhangs, right? So you have complementary bases kind of just sticking out. And so in theory, if you think about it, if this, zoom in even more, if this has some complementary bases to this, to this down here, these two pieces, they're floating around in a tube. They have a very good chance of resealing because they're complementary, right? So this right here has a complementary base pair. So these two edges have complementary base pairs here. So this definitely can re-anneal in the tube. That can happen. The same is true for over here. Those two edges, they can re-anneal in that tube. And it's possible. So when you're doing cloning, this is one of the things is in theory, this is what you want. In reality, it's like Instagram versus reality. Instagram, this is what you get. Reality, you get a bunch of things that maybe you're not showing or you don't think about, but these can seal up, and hopefully you have at least a couple singular isolated GFP genes because you're only interested in that. Okay, so yes, that is isolation of the GFP gene. You're going to do it via restriction digest. You're going to figure out the names of those enzymes, but in theory, this is hopefully what you get, is that this turns into isolated GFP. Okay, so I saw a couple other people writing this. Let me scroll back up. I see something about a plasmid vector. Okay, was linearized. Okay, great. That is the formal terminology. linearize the vector. And the vector in this experiment, some people wrote it, but I'm giving you an opportunity one more time. If you didn't write it and if you were vague in the beginning, here's your opportunity to be specific. What is the vector that we're using? Great. Yes. Cool. So the vector that I have chosen to do this experiment is a p-bad. It's called p-bad. It is a plasmid, and a plasmid is a type of vector. So this p-bad vector has an origin of replication. So if you do, and I'm giving a little bit of hints away, if you do get it into your host cells, the host replication machinery will recognize the origin of replication and will also replicate that plasmid. And so remember that plasmids are circular. Why do we use circular plasmids when we're doing cloning? Because typically you're going to be using a prokaryotic host, and prokaryotic chromosomes are circular. So you're sticking in something that's the same shape as the host's genome. If you stuck in a linear piece of DNA, the host recognizes it as a pathogen and destroys it. You don't want it to be destroyed. You want it to be copied. Okay. So the way that you linearize the p-bad vector, lo and behold, is the same exact way using a restriction digest. So this is a circular piece of double-stranded DNA. And so if I just drew one tiny little cartoon, I'm just going to draw the cartoon underneath the writing. So you have, okay, this is hard. Yeah. Okay, you have this circular piece of DNA, right? Your little hydrogen bonds all the way. Okay, so you're going to use those same exact enzymes. And here's the reason why, because remember what the goal is to do next, or basically what you want to do is you want to get this GFP gene and you want to kind of stick it into this circle. You want to make circle, can you scooch over, let me wiggle my way in here. And now we're going to stick it into the DNA. So the only way that you can do that is if you can complementary, not that one, this one. If you can complementary base pair these edges to the plasmid, that's a really good way to do that. So those specific edges are called sticky overhangs, because and the overhang sequence is the result of the enzyme you use. So if this is the sticky overhang for restriction enzyme number 1, if you use restriction enzyme number 1 over here, you find a place in there that you can use that same restriction enzyme, that is going to create the complementary overhangs. Cool. So if you can find one, if you can hopefully use the other enzyme that you use to isolate the GFP, what this is going to create is, this is now going to really be hard for me to draw, but I'm going to do it for you. Okay. All right, so you've created those sticky ends, right? So here's your base pairs that are just dying to base pair with something. And then here's your other overhang, your sticky overhang, base pairs dying to base pair with something. And then you have that little chunk that's kind of floating around too. Plus, okay, these ones I'll just draw with purple there. Great. So hopefully what you can see, maybe if I copy and paste this, we'll do it in the next one, okay? So what you can see here is that this, once you cut it, there is bases that are just, it's looking for its complementary bases so that it can base pair together and, you know, reform this nice chunk of DNA. So if we're thinking about it in terms of a tube again, this is all happening in a microcentrifuge tube where you've put copies of the p-bad vector. You don't just have one circle, you have, I don't know, hundreds of circles in here. Bunch of p-bad copies of p-bad vector. And then you add your restriction enzymes, number 1 and number 2. And then you add that into the tube and then you let them do their job. And then hopefully what you're getting inside this microcentrifuge tube is a bunch of opened up or linearized P-bad vectors with the little chunks floating around too. Okay, so that's what you're hoping to get. And yeah, the same way that this could reseal with its original chunks of DNA, this can come over here and reseal and make a circle again. And that will happen to some of them. So you're starting with this, you're adding enzymes. In theory, the enzymes are going to cut perfectly, and then that fragment is going to remain. Those two fragments will remain separate. In reality, it can reseal to form a circle again, or perhaps only one enzyme cut, or perhaps no enzymes cut. So in reality, what you have, so this is in theory what you want, and in reality, what you have is the plasmids have resealed. You also have plasmids that were only cut with one restriction enzyme. Okay, so they don't have, let's say they don't have the purple sticky end. So how are you going to get this in here? You can't, because this will not be complementary to the other side. Okay, or no restriction enzymes cut. Those are all options that can happen and that can exist in that too. Okay, but the take-home message here is the two main steps that need to be done is isolating the GFP gene, creating those sticky overhangs, creating those same complementary sticky overhangs by linearizing some sort of vector. So in this case, we're using a plasmid vector. Okay, so now that we have that in theory, these are, let's circle, this is what we want. This is what we want. Recall that there's other things that are possible. Okay, so let's go with number 3. Don't press enter, just tell me what has to happen next, the next major step. All right, enter. Okay, so we've got a little, again, we have a couple people that we're getting ahead of ourselves, but I see some right answers. Great, yes, okay. So remember, we have 2 tubes with theoretical linearized vector with theoretical isolated GFP gene. And so the goal here is to create not just any recombinant DNA, but the recombinant where you have GFP that GFP that has been inserted into P-bad. Okay, and I think in the video I said if you do create this recombinant plasmid, we're going to call this recombinant plasmid P-glow. So what you're hoping to create is here's your P-bad backbone. Okay. Right, and then you're going to stick in, hopefully, by combining the 2 DNAs together in the tube, your GFP into that thing. Now, why did that work? Because the sticky ends stuck together because they're complementary. That is the goal. This is your P-glow. Okay. But if you think about it, any combination can work because you're just taking your pipette and you're taking a little sample of this, a little sample of this, and you are combining it into one microcentrifuge tube. So there's no way for you to just get the linearized PBAD. There's no way for you to just get the isolated GFP. You have all of the contents and you're just hoping that the correct combination, which is why we call it a recombinant DNA, you're hoping that that occurs. Okay, so this right here is an example of a recombinant piece of DNA. In this example, because it's a plasmid, we call it a recombinant plasmid. But the definition of recombinant is any two foreign pieces of DNA being sealed together that normally don't get sealed together. Okay, yeah, and in order to do that, let's see, I see it. This whole thing is called a ligate, I guess that's the approach, is a ligation. Okay, good. And what is the most important, and some people have it, but I'm going to give everyone a chance. What is the most important thing, reagent, that is required for a ligation? And the hint is in the word, in the name of the approach. Yes. Okay. This is going to require ligase. The same old good old ligase that seals the final phosphoester bond is the same ligase, because this right here, it's complementary. Can I zoom in even more? It's complementary, but it just allows the two strands to hydrogen bond. But the backbone still is missing one phosphoester bond. These are going to just allow the two strands to hydrogen bond. So they're going to be in there, but it's kind of like loose. So you got to make sure that you seal that backbone so it's nice and covalently bound, right? Hydrogen bonds are not covalent bonds. They can easily be broken. Covalent bonds is going to be better, okay? Okay. Alrighty. So that is what is going to happen next. So now we're still in a microcentrifuge tube, and to the eye, this is still clear liquid. And so the question, so Ivan is asking, what is ligase? Ligase is an enzyme, and it's an enzyme that is used in DNA replication, and it's used in the termination process of replication, which was covered on the exam. So hopefully you didn't forget that quite yet. Okay. So great. Yes, it reseals the backbone by catalyzing a phosphoester bond. Stephen's coming in, giving everyone the answer. The first restriction enzyme is EcoR1. You are correct. So we can put that there, right? I knew you couldn't let Tuesday be better than you guys. Okay, good. All right. So here's where we're at. In theory, we have a recombinant. We have a recombinant plasmid. Okay. So next major step. What is the next major step that what do we do with this ligated product? So don't press enter, just go ahead and type. Okay, enter. Great. Yep. Nice job. Okay. Couple you are still getting a little bit ahead of ourselves. We cannot do PCR on the ligated product yet, or there's no need to. Okay. So once you make this, remember this, the overall goal of molecular cloning is to make bacteria, force them to make for you tons of copies of a certain gene, and then you pull it out of them, and then you use it to do experiments. So far we haven't even talked about bacteria. So we need to, the next step, as most of you are saying, is we need to transform, just like when we were talking about the transforming principle and how DNA can transfer into certain hosts. That's the same idea, taking things that happen in nature, using it for science for our benefit. So yes, the thing that we're wanting to do is we want to insert the ligation product, or to insert the, hopefully the P-glow, that's the real goal, to insert the P-glow into the host organism. And in this case, if you didn't tell me what the host organism was, please tell me now. Yep. So it is a strain of E. coli called MM294. Okay. So you're trying to get this P-glow, recombinant plasmid into E. coli. What has to be special about these E. coli? Because as far as I know, E. coli are an organism that exists out there in the world. They don't really like to just take any old DNA. So what do you have to do to these E. coli before you use them? Yeah. Yes. And so these E. coli need to be competent. And in parentheses, how do we make them competent? Thank you, Ashley. Calcium fluoride treatment. Okay. So this was already done before. So you got the E. coli cells, you treated them with calcium fluoride. The calcium chloride kind of coats them in a positive charge so that when you do surround them with negative DNA, the negative DNA is attracted to them. You do a couple other things that kind of like disrupts the integrity of the cell wall and the cell membrane. So you have a better chance of getting foreign DNA into them. Because if you just had your regular old E. coli from, like, let's say you somehow took it out of your gut, they're not going to accept this recombinant plasmid. Very few of them, you're not going to get that recombinant plasmid into these bacteria. So you have to kind of like puncture holes in their membrane and their cell wall and then coat it in a positive charge so that the negatively charged DNA can be attracted to it. Okay. So that is the goal. The approach is called a transformation. And there's no special reagents that help the transformation happen more or less, except for heat and cold. So you heat shock them, and so heat disrupts things, right? Degrades things. So the heat is basically going to make the membrane even open up more holes in the membrane. And then the DNA can whoop, kind of just jump on in, and in the cold helps heal them up a little bit, but the cold. So we're basically torturing these little babies. Okay, so transformation is the process by which you try to insert your desired recombinant DNA into a host that has been previously made competent. You can make them competent by calcium chloride or electric shock. We did not do electric shock, mostly because when we do it in person, I don't trust you guys with electricity. So, and liquid, so we use calcium chloride. Okay, and so essentially, the goal here, again, this is all still done in those tiny little plastic, the tiny little plastic microcentrifuge tubes. So we're still in a tube. But if we zoomed in and looked at that, now we have a bunch of bacteria. So E. coli are kind of like this pill shape, and they have, you know, their own host chromosome. It's really giant. And so hopefully you don't just have one E. coli in the tube, you have a bunch of them. So here's all your little E. coli cells, right? More than 4 too. All right, and so you're hoping to get this tiny little plasmid into the bacteria. And in theory, if you did everything right, and if science was wonderful and perfect, then that's what you would have. You would have your bacterial cell with the plasmid inside. And remember that plasmid has an origin of replication. So when that bacterial cell goes to replicate its own circular chromosome, the machinery is like, oh, there's another origin. I'll go do that one. You guys do this one. I'll do that one over there. We'll replicate the DNA, and then the bacteria will split. And now this bacteria has the copied circular chromosome and the little plasmid. And it just keeps going and going, right? But if we're still in a tube, one tube of bacteria is not going to give us enough product to do 5 years worth of experiments, right? And this is a very small space. So you're going to run out of space. So once you have transformed your cells and you've let them kind of heal or recover, so it's transformation followed by recovery. Okay. So you're like, please forgive me. Here's some food. Here's a nice, comfy, warm temperature that you love. Please heal up your cell membrane and your cell wall and just keep that plasmid inside and please be happy. Okay, so now we're gonna move on to step number 5. So recall that this is what you have. You have all of your cells. Some of them will get the plasmid. Some of them will not. Sometimes the plasmid gets in and the bacteria is like, nope, please exit. Sometimes you don't insert this recombinant plasmid. Remember, any combination of these things can exist because you put all of that into a tube. So there's a lot of different options for what you're inserting into a bacteria. And so now you've just increased your possible options. So we have to come up with a way to kind of get rid of the bacteria that potentially got the inserts that we're not interested in. So if you got a resealed plasmid back into if you got it into the bacteria, we don't want that. That plasmid doesn't have our GFP. So let's get rid of that. We need a way to screen that out. If we got some weirdo combination where it was like GFP and some other chunk and we got that into the bacteria, we don't want that. So we need a way to screen that out. So we need a way that will allow us to choose. So how to choose or kind of kill off the other bacteria that we don't want. So we want to choose the E. coli that only has been transformed with the GFP that was inserted into the P-bad vector, otherwise known as P-glow. Okay, that's what we're hoping to do next. So how do you do that? What is the next major step that we do? So we're going to do something with the contents, the transformation products. We're going to do something with those transformed E. coli. So type, give me a second and give your classmates a second, and I will tell you when to press enter. I'm going to have water. Okay, go ahead and press enter. Okay. Okay, so I see some, a good start. So we need some more specific. So people are saying you're gonna plate them in different conditions. Okay, what are some of the different conditions? And just to let you know, this is called selective growth media or selective growing conditions. Okay, let's see what you all are putting. Okay, so yeah, so let's go ahead and write some of this stuff here. So you're gonna take a small little portion of these bacteria that were transformed and you're gonna put them in a variety of different plates. because the goal here is to select for the E. coli that were transformed with P glow only and basically kill off any other option. So the one thing that if you're going to grow bacteria, you need to give them the nutrients that they need so that they can make ATP, so that they can do DNA replication, and that they can do cell mitosis, and that they can divide. So no matter what, all plates at a minimum have what's called Luria bertani broth, or LB. Okay, all of them. If you don't put LB, the bacteria cannot do anything. They'll die. They won't be able to make ATP. They can't do any cell processes. So at a minimum, Luria bertani broth is on all of the plates. Okay, so this is the minimum nutrients so that the cells can make ATP. Yes, like it contains sugars so that they can break them down and do glycolysis and et cetera, oxidative phosphorylation, ATP. Okay, and so other people are saying that some of the plates will have ampicillin. And this is true. And some of the questions on the worksheet asked you to kind of think about, why would a scientist need to add ampicillin? What kind of bacteria would it kill? So it better not kill these bacteria. If it doesn't kill them, what do these bacteria have that allow them to live in the presence of ampicillin? You don't have to answer now, but this is why I ask those questions on the worksheet. So ampicillin is one of the ways to get rid of the extra bacteria we don't want, right? Because ampicillin is an antibiotic, so it should kill most bacteria, unless you have a special little gene that protects you from that, okay? Another thing that people are saying that we need to put on some of the plates is arabinose. You can tell by the suffix that arabinose is a type of sugar. So this sugar is going to provide some sort of clue for the scientist. So it does something normal in nature, and then we take that idea and we're like, oh, I know, we could use that to somehow get rid of some of the bacteria we don't want. Okay, cool. So this allows you to select, and then this allows you to do what's called screening. Okay. And then let's see, did anyone else put anything? Agarose? No. Okay. Some contain both of those ingredients, right? So when you're looking at plates with and without arabinose, you're going to be looking at plates under UV and not under UV. Now, in this case, we're not using UV to induce skin cancer. It has nothing to do with skin cancer. UV is the wavelength of light that excites GFP, the protein product. So the protein product doesn't just naturally glow green. The protein product needs to be excited by UV light wavelengths. That's when it glows green. So that is going to allow you to see, so UV excites GFP protein, so the protein version, not the gene. So if the DNA hasn't been translated into a protein, you can shine all the UV light you want on the DNA and it's not going to make it glow. It's just going to cause pyrimidine binders. So it only excites the GFP protein. And so when it excites the GFP protein, this is what causes the glowing. So anytime you see glowing, that tells you that those cells took up the GFP gene, transcribed it, translated it into the right structure and to the right function so that when you do shine UV light, it glows. So glowing means functional GFP, which indicates successful transformation. Okay, and so hopefully there's a lot of different combinations and we're not going to talk about them here in class, but let's say this is your Petri dish. No, I'm not a cartoonist, but let's say this is your Petri dish and you're shining your UV light. So UV light is like, it looks pink, right? And what you see is that some of your colonies, so these dots are not single cells, they are hundreds of cells that derived from one cell overnight, that one cell divided into 2, into 4, into 8, into 16, etc. Until you come back the next day and it's a little clump of cells called a colony. Some of these colonies are green, indicating that they have functional GFP protein, and some of them, are not green. So they have something that allows them to grow on this plate, but they did not make functional GFP. Okay, so moving on from there, this is a give me question. Which of those 2 colonies do you want to proceed with, or did I proceed with? The green ones or the non-green ones? Yes, just making sure that we're all on the same page. Yeah, so we want to make sure that we pick the colonies that are glowing green. Sometimes you just pick one colony, sometimes you pick a couple, depends on how big your colonies are. So in this, I think in this lab, I picked 4 colonies, okay? And so now we can proceed to the next major step. So with your eyeball, you can see like, hey, these cells have functioning GFP, but scientists like to ask the same question and answer it multiple different ways. So we want to confirm, we're like, okay, yeah, we see that the protein is glowing, which means we see that means that the protein is folded properly, but how do we know that the gene is still the same sequence that we started with? We want to really make sure we do that. We want to really make sure that the sequence hasn't changed in all of these processes, because we know how often mutations can happen. We only want to stick in this original GFP sequence. So type, don't press enter. What do we do next? Okay, so the goal here is to confirm the sequence of the GFP gene. Okay, go ahead and press enter. Yes, right? And yes, there is a connection. Remember at the beginning, if you watch my intro video, who I said my favorite musical artist, Snoop Dogg. Okay, so yes, there's a connection. All right, so yeah, if you want to confirm the sequence of a GFP gene, these cells, I'm gonna go ahead, where's my little picture here? You're assuming these cells, based on their glowingness and based on the ability for them to grow in the presence of certain reagents, you are assuming they not only have the PBAD backbone, right? So there's certain genes on this PBAD backbone, but it also has the GFP gene. And so you want to make sure that that thing I just highlighted, that sequence hasn't changed. So in order to do that, you are going to do, and people are saying this, you're going to do a colony PCR, right? So that's one of the things of PCR. You can confirm, this is kind of like a genotype. You're confirming whether or not the gene is there, yes or no. So in order to do that, you design primers to either side of your region of interest. Your region of interest in this scenario is your GFP gene. So the scientist, you're gonna do this using SnapGene. You are going to design primers to this GFP gene. And the GFP gene, if the gene is there, you're gonna get a product. If the gene is not there, then you're not amplifying anything because the primers won't be able to stick to anything. So what do you do next? After PCR, you have one of 2 options. So if you are looking for deletions or insertions, you would run a gel electrophoresis. But that doesn't tell you the actual sequence, the letters of the nucleotide. It just tells you, hey, it's shorter or longer than what you thought it should be. That's not the information we need. We want the actual order of all of the nucleotides. So what do you do instead? So you do your PCR. What do you do with the PCR product? Yes, whoever just answered, Steph, Sierra, and Cole, your answer is collectively correct. So yes, what do? You take it, so colony PCR happens in a tiny little tube. You take a sample of that product. Yes, you send it to a sequencing company. So yes, you send it to a company and they perform sequencing on it. And then they send you the results via email. And then you compare that sequence to the known sequence of GFP. So you compare your sample to known GFP sequence. And the sequencing will be sent to you as a chromatogram. And that's that picture with each curve is a different letter that signifies what base that probably is. And yeah, you compare the sequences. And if the sequences are perfect and no letter is different, then your cloning has been successful. If there's one mutate or one difference or two differences, you got to start over. And that's the nature of cloning. So yeah, that's what you're going to do here. So what are the desired results? The desired results is that your insert, so your GFP insert, is the same as your GFP sequence that you started with. Okay, assuming that that's true, now you can proceed. So remember, we only have a tiny little plate, and here's the cool thing. So when you take these little colonies, basically you're taking these little colonies, and I'll draw a little, an arrow. You're just scraping them off the plate, and you're sticking them into a tube and just kind of sticking them into the buffer. That's all you have to do. And so what happens is you're scraping them off, so they're gone. You stick this plate back in 37 degrees. And the cool thing about bacteria for science is that there's no way you as a human can get every single cell off. So even though it looks clear, I don't see any green spots on this plate anymore. You stick it back into the 37 degrees. You come back the next day. You come back. So they're there again. So it's really cool. And that's why it's important to make a circle around where you scraped, because then you know where to expect the bacteria to be the next day, which is awesome. As long as there's still nutrients on that plate and they haven't used them all up, you can still do that. So that makes it really important, because the next thing that you're going to do is once they reappear, you want, now you're ready. Now you're ready to bulk up and say like, hey, I transformed my bacteria. I got rid of the crappy bacteria with inserts that I don't care about. I confirmed the sequence of the GFP. It is correct. Now I can grow this up in humongous quantities so that I can start doing my experiments. This is all just to do experiments, right? So what do you do so that you can bulk up? So the goal is to make a large quantity of E. coli that have been transformed with P-Glow. So type. Amazing. See how cool science is? Okay, so type. What do you do so that you can make the large quantity of E. coli? Don't press enter yet. Okay, enter. What happened? Enter. I'm gonna let a couple more people go for it. Close, close, close. We already PCR'd it. But I get where you're going with this. Okay, last guesses, even if the guesses are incorrect, because so far no one is correct, so just go for it. Okay, yeah, kind of, you're gonna make an E. coli farm. Okay, so this, if you think about it, these tiny little speckles, that's still not enough to do 2 to 3 to 5 years worth of experiments. Those are maybe like, I don't know, 1,000 cells or something like that. No, we need to make like, we're talking millions. We need millions of bacteria cells. So right now they're on a solid media. They're on a Petri dish. It looks like yellow, pale color yellow jello. It's nasty and it stinks because the E. coli stinks. Okay, we don't want them on a plate. So in order to grow like a mass amount, we don't want like giant, huge plates. Instead, bacteria also grow in liquid. So what we can do is we can take a giant flask, like an Erlenmeyer flask that you've seen many times in the lab, in person or on paper like this. And instead of it being a solid media, you still give them the minimum nutrients that they love and they need to make ATP. And then you still add the selective reagent, which is ampicillin. You still add the arabinose. Actually, this is a maybe. You don't have to add it because arabinose is not going to allow them to make the product or not. It's just like it makes the GFP grow or not. So we'll put that in parentheses for now. But essentially, I'm going to draw this in a different arrow color. How about brown? So you take those same colonies, you scrape them one more time, and this time you put them into a liquid culture. Okay. And then you let them grow overnight. So to make a large quantity, it's called inoculate. Inoculate the confirmed colonies. You've confirmed them with PCR. Okay, and you inoculate them into liquid selective media. And then you let them grow overnight at their most favorite temperature, which is about 98 degrees Fahrenheit. It's a little too warm for me, but they love it, which is why they love to grow inside your gut, because it's 98 degrees in there. And you let them grow overnight. So this media, when you first pour it, it looks like clear lemonade, okay? If the bacteria grow, you come back the next morning, now it's cloudy lemonade. So anytime you go into your fridge and things start looking cloudy, now you know that it's bacteria that is making it cloudy. When things start getting fizzy, like I don't know if anyone has ever left OJ in the fridge for too long and it starts like, wow, I didn't know this was carbonated orange juice. That's because the lid is on really tight. Sometimes the bottle puffs up. That's because the bacteria have run out of oxygen, but they're still eating the sugar of the orange juice. So they're doing fermentation. Yeah, no, not hummus. Okay, so now you have a big vat. There's a bunch of bacteria that you know originated from 4 tiny little colonies. All those colonies have been previously confirmed. They have the correct sequence of GFP. So now you have, I should be drawing them in green. So let's pretend these are all green dots, okay? Because they have the GFP, if you shined UV light on them and they were grown in arabinose, that whole entire liquid would be glowing green, like ecto cooler. So, all right. And so now you have this big vat of bacteria with making the protein product, but you don't want to do your aims with bacteria. You just want the protein. Okay. So the next and final major step of the preliminary experiment is to isolate just the GFP from your cells. Okay, and you leave the cell components behind, you leave the PBAD behind, you leave everything behind. So what your goal is, here's your goal product. Nope, this one. You want pure GFP protein solution. No other proteins, no other vectors, no other cell walls, cell membranes, no nothing, just protein in buffer. So this one's an easy one. We don't need to wait to make, to press enter. So what, and it comes from a homework assignment question. What is it? What do we have to do? Yes, we have to do high C. So we can make our ecto cooler. Do you guys know ecto cooler from, did you ever drink that as a kid? Ecto cooler? Anyone? No? Okay, I'll take it as a no. Well, for anyone that wants to know what ecto cooler is, it's a type of high C. You all know what high C is, the drink, yeah? Okay. It's a type of high C, a flavor of high C with the little green goblin from the Ghostbusters. So it was called ecto cooler and it tasted like OJ, but it was green and it was so awesome. So yeah, so you do hydrophobic chromatography, yeah. Okay, and so that, I know it was long, but this is the preliminary experiment, and that was why we did it in class, to make sure that we kind of all can understand what, I mean, molecular cloning is done like, I don't know, all the time in a source, in a series of like 2 to 3 days, and this process gets repeated over and over and over. Because there's many times you're like, nope, guess I didn't transform it, better start from the beginning. Oh, nope, guess I have a mutation, better start from the beginning. So you have to do that a lot. Okay, so any questions on any of the steps from the preliminary experiment? Okay, awesome. Well, that's what I wanted to cover today. Now I will open it up to any questions about the data, proposals in general, background, what you have to do for AIMS, anything. Okay, cool. So just reminding you that, let's see, I'm gonna stop this share, go through my 300 tabs that I have open. Is this what I wanna see? No, I want to see the next week. Give me a second. I can go ahead and stop the recording too.